Okay, great. So I am so pleased to be here and for this to be part of this webinar series. I'm going to tell you a bit today about some of the work my lab does in multiscale modeling of tissue remodeling and damage. We currently look at several different organ systems, and that's kind of how we characterize the different subteams in my lab. But all of the problems we look at involve some aspects of this balance between tissue degradation and tissue formation that in many tissues is part of normal homeostasis. So we might be continuously turning over our tissues for and remodeling them for healthy tissue maintenance. But in some diseases, we can have dysregulation in that process, and this balance can be tipped towards a net tissue formation, for example, in cancer, or it could be tipped in the opposite direction towards a net tissue degradation, and that's the case in generally osteoporotic bone diseases. And we're looking at different kinds of chemical factors. These could be hormones, drugs, foods, cytokines, toxins that could either initiate the progression of the disease state. And so we're thinking about from healthy towards the disease phenotype, or that could positively modulate the disease state back towards a healthy phenotype. And so we're thinking about foods could be in both contexts, right? Glucose dysregulation in diabetes is from food, and that's more of a pathogenic one. And then we have things like dietary supplements that could be positive mediators for bone uh, regeneration. And so we're thinking about all of these different things in the back of our minds when we approach different problems. I won't tell you about all of the different areas that we're working on in the lab right now, but I will give you a little taste of what we have accomplished and what are some of the active things we're doing right now. And I am an associate professor in chemical and biological engineering at the University at Buffalo, which is part of the State University of New York system. So the important people in my lab are our students, trainees. So this is the picture as of the 1st of August, so 2 postdocs and 3 undergraduates. The balance are graduate students. And we have generous support by NSF and NIH and the State University of New York system right now to support the work that I'm going to talk about today. And so in general, my lab builds models for 2 types of problems. One is kinetics, and so we're thinking about species that it could include biomolecules or entire cells or even entire organisms. We have some published work related to bumblebees where we considered the transitions of bumblebees. And in all of these, we're thinking about the rates at which they react or interact or maybe changing state in some sort of biochemical networks. So the example snapshots here, looking at a reaction network where an ACE inhibitor drug is working on this system, renin-angiotensin system. The second is an ODE-based model related to cancer cells and their populations, along with some proteases that are changing the extracellular matrix environment through which those tumor cells move. And then the other area of our lab focuses on mass transport. So some of the work relates to small molecules like glucose, but other macromolecules, many proteins or therapeutics, and the cells that are moving through these heterogeneous porous biomaterials, but often our biomaterials that we look at are also dynamically changing in response to the reactions with the different species that are in the system. So proteases remodeling tumor microenvironment. In the snapshots below, I have a picture of a cumulative release of a drug from an advanced biomaterial that can chemically degrade, and it has some heterogeneities in that process that leads to some of the different sizes. The colors represent different sizes of particles, drug release particles, and some of them can have different complex phenomena where larger ones may be able to release faster than smaller ones under certain circumstances. The middle vignette is for our cancer metastasis work, and then the final vignette is a picture for membrane transport for our kidney work, and I'll tell you a bit more about the kidney work later today. And so we use a wide range of methods in the lab. They're all computational. So we look to the specific problem and say, what kinds of tools do we need to answer these questions? So the common wheelhouse of tools that we use include differential equations, and these can be ordinary or partial differential equations. And we use a range of different numerical methods as appropriate. We have experience doing a lot of programming and graphical user interface development, particularly for collaborators or for educational purposes. More recently, we've been doing a lot of finite element modeling, both in fluid dynamics and also in the structural biomechanics. We do some dynamic systems and data analysis work. And then in the last 8 or so years, we've been doing quite a bit of agent based modeling with different types of agent based software platforms. And you'll see some of that work today as well. And so these are the areas, the diseases and biomedical topics that we've been publishing on or actively working on through funded projects in my lab right now. So you will hear a bit about the diabetic kidney disease story today. I won't have time to tell you about cancer metastasis, but have a few publications on that work. I will tell you about our gut bone axis work. Immunology and immunotherapy has been an area where we started with tuberculosis, and then we've moved to several different things throughout the lung, and more recently with COVID. So you'll hear a little bit about the COVID and lung fibrosis story today. And very recently, we have an R01 funded on some advanced drug delivery devices targeting the eye, the back of the eye. But I won't have time to show you any of those preliminary results for that modeling effort. So currently, my lab is looking at bone, lung, kidney, eye, and cancers. And this is sort of how I organize the talk. Again, we won't have time to visit the cancer or the eye stories in the lab. So, the first story is about the kidney. And diabetic kidney disease modeling is the area in my lab that has been supported the longest, not just by the NSF career, but by several other funding mechanisms in advance of that. So, this is the one that I started as a first year assistant professor. And we've had all the generations of my lab have been involved in this project in the team. And so I'm showing sort of oldest to newest, the individuals in the lab who've contributed to this work so far as being circled there. And so we have several publications on several different aspects of this, but this really has been a core emphasis area for our team. So we consider different kidney cell types. So we specifically look at a region of the kidney called the glomerulus, and this is where blood gets filtered. And we think about how these glomerular cell types are damaged in diabetic kidney disease and then how they interact with each other. So this illustration in the top corner is representing a glomerulus. So we have blood vessels that come in, branch into a network of capillaries, and then leave. And the blood is moving in the capillaries, and things that are getting filtered to collect and form urine pass across these cells called the podocytes and are collected by this outer circular bulb, more a spherical bulb. And then if we look at the actual labeling of these cells, we have the blue cells are the mesangial cells that hold together this bundle of capillaries. The red circles and ovals are cross-sectional views of those capillaries, and they are lined by capillary endothelial cells, and then they are covered by this green layer, this basement membrane, and surrounded on the outside by this network of podocyte cells. These podocyte cells fit together with what they call foot processes, and that's what's indicated by the small sort of diamond shapes on the outside. We can have interactions in each of these rectangular boxes. So there can be interactions biochemically and biophysically between the endothelial cells and the mesangial cells. So you have the blood is inside of these kind of oval and circular areas, and nutrients can pass between the endothelium into the mesangium, and so that's an important set of crosstalk between these two cell types. There's also crosstalk, I'll point to this top one, from the materials, the blood that's inside the vessel being filtered, through the endothelial layer, the basement membrane, and through the gaps between the podocytes. So we look at that kind of interaction, but then we also look at this region where there can be all three of these types of cells in close contact in some areas anatomically. And so we think about this is the general filtration path that most people think of, but we think of these other interaction pathways as well. And we're thinking about the different kinds of chemical signals that are passed, forces that interact that then change these microenvironments. One of the key things that happens is these mesangial cells can grow and start to um, push on the blood vessels. And so there's a lot of biomechanics going on. The entire If the inside is being stretched out and pushing the blood vessels, the podocytes wrapped around the outside also get stretched out. And so we're just now starting to incorporate these biomechanical aspects, but we've been building on the biochemical aspects for about a decade now. So first approach this by thinking about hyperglycemia and the filtration barrier, specifically looking at podocytes first. And so this is a view of looking inside the blood vessel. The yellow are representing molecules, small molecules and proteins that are inside of the blood vessel. Normally, small molecules only should be able to pass through the glomerular basement membrane and through the gaps between the podocytes and their foot processes. In long-term exposure to elevated glucose, there are effects where the foot processes start to peel off of the surface, and some can die or just be completely detached. And so that changes and disrupts the filtration barrier completely. Some of the podocytes, we don't represent this graphically here, but some can stretch and cover more surface area, but they're working harder. And so then they are more susceptible to becoming to failing after they've been overworked. And so this all changes and disrupts the ways that proteins can pass through these membranes, and you'll start to detect protein in the urine. And so we're very interested in this pathophysiology about how the onset and the progression occurs, and being able to predict that evolution in advance of severe protein loss in the urine. And so as a team, we've been building submodels for these different glomerular components and how the different biochemical networks interact. So, how glucose interacts with key molecules in podocytes, for example, angiotensin 2, how it may interact with nephrin, other molecules of the mesangial cells that regulate its growth and behavior. So, we've had several publications related to the biochemical networks in isolation. We've also done some work with the transport, this movement between these different cells and those communication. And we've also been building models related to the entire tissue structure and crosstalk. So our first agent-based modeling approach was really to think about how the connections between the podocytes might fluctuate. This particular model didn't allow us to have the biochemical aspect and really have the layers growing in the way that could account for the forces. And so these, I would say, is sort of um, the rudimentary version of connecting these glomerular components into a multi-scale framework. But this was the vision as we got the career award started and as my first two PhD students were graduating. As I moved to Buffalo 2.5 years ago, several new students have joined the team or had joined already before moving and have really come into their own. So three PhD students are currently working on the kidney project. One of them is Krutika Paditar, and she is looking at in the the blood vessel, we can have interactions between the glomerular endothelial cells and cells that are uh, part of the immune system, so specifically macrophage cells. And so we know that diabetes is related to chronic inflammation, or those are chronic inflammation is a state that many diabetic patients experience. And so there have been studies showing that macrophages do infiltrate into the glomeruli, and that um, there is crosstalk between macrophages and endothelial cells, and so we've been looking at cell culture studies of both of those that have been published. So we don't do experiments in my lab, but we're trying to build reaction network models that can take glucose and inflammatory stimuli and involve the crosstalk between these two different cell types. And so Krutika's work has focused on logic-based modeling technique to simplify the signaling processes between these endothelial cells and the immune macrophages, and really coming up with some nice work that can predict the things that people have measured, these different cytokines, but also phenotypical changes like the gap width between different endothelial cells. And so that's ongoing work. That we aim to publish this year, but then she's had a 6-month co-op in industry, so we should be submitting that pretty soon after she returns from her internship. Haryana is another PhD student in the lab, Haryana Thomas. And so last year in the Foundation of Systems Biology and Engineering conference, he had a paper about the different fiber fractions in the mesangial zone and how that modulated the signaling distance that was then available for molecules in that environment to communicate with other cell types. More recently, he's been working on a model about glucose and its interactions with collagen remodeling in the mesangial space. It involves some different immune cell types as well as the mesangial cell types, and has been uh, parameterizing this with published studies. And it's, again, very near completion, but he also had a summer internship and will be returning soon. So we've pre-printed this work, and I believe Critica's work as well, but those are ongoing projects that we hope to publish this year. Nick just finished his master's degree in May, and so master's thesis has been submitted on this topic, and then he's continuing as a PhD student in the lab. So his work really lets us now connect these submodels for damage in each of these tissue layers. So we have a finite element code that we can then use to study the biomechanics and the fluid dynamics, these transport processes through the tissue, and we want to allow the transport properties to evolve based on those chemical interactions. And we're thinking about different issues with the time scales involved in these processes, whether the simulations need to be distinct or whether we can simulate different timescales in different simulation environments and then reconnect them. So we're working on exploring some of those areas. But the main idea is that we're still thinking about this uh, glomerular filtration barrier and zooming in from our early work, and we're representing this space between two podocyte cells and a small region of glomerular basement membrane in the space between two adjacent endothelial cells. And this is his rendering in the finite element software, where these colored areas are actually the spaces between these cells or the space that is through this basement membrane. So the endothelial cells would be here on the left and the right, and then the podocyte cells, you see the outline of their shapes here on the left and right. So this is a flow domain in the capillary. This is the small channel between the two endothelial cells. Yeah, so misspoke. These would be the endothelial cells, not here. This would be just bulk flow. So two endothelial cells here, the small channel between them. We have a biphasic material representing the membrane. Then we have a small gap between the podocytes representing the space between those two podocytes that usually has a mesh of nephrin and other molecules there, and then the free space where the molecules are collected. So we are modeling the mass transport through this domain, we're modeling the pressure fields, velocities, et cetera, and then we want to be able to couple the concentrations of these different species to the material properties that then will allow, right now, small molecules, water, glucose can move through this environment, but we want to be able to change the material properties dynamically in a way that would signal the damage and then would then enable large molecules to be able to penetrate at later stages. And so Nick has been working on some really detailed meshing and other finite element work on the anatomy to have high fidelity and grid points and things like that was a major focus of his master's thesis. And then this is showing the kind of first three-dimensional attempt where we have the flow channels having high pressure between the podocyte slits, and then working on various other aspects of this. So this is really more of the velocity and pressure fields, but we can also turn on the mass transport, and then we're starting to work on the infrastructure to be able to couple these to our previous models in MATLAB. So, shifting gears, and think about the lung work for a bit. And this is just a subset of the things we've worked on in the lung, but this is the most recent story. And so, postdoc Mohammed Amin al Islam in my lab has been the major person in my lab working on this project. But we've been part of a large team called the SARS-CoV-2 Tissue Simulation Coalition. This is a team led by Paul Macklin in Indiana, bringing together people who are experts in mathematical modeling, systems biology, but also cancer and immune science, as well as sort of computational tools. And so it's a wide ranging group of people, but it's really been a pleasure working together. So mainly the idea has been to develop a lung tissue simulator, and it's the blue is representing the alveolar surface of the lung epithelium, and we're thinking about single layer thickness of this epithelial tissue. This is in three dimensions, but we've just represented circles here, but it's layer thick spheres. Um, we are thinking about the infection, and we have different immune cell types as well as um, different kinds of chemical factors. So we don't visually represent the chemical fields generally on these cellular fields. And then but we have them. And then we also have virions in the background, but they are not discretely modeled. They are also a continuous field. And so in the tissue, if we think about this collection of these circular, in our representation, epithelial cells, we consider the virions getting into each of the cells, a complex network of replication and other ways that the virions survive, reproduce, and disseminate. And another subgroup on the team really focused on this. So this was not my contribution to this work, but we do have several people on the team whose entire research programs focus on viral replication and relevant related viruses like influenza. And so they have really brought their expertise to bear on this aspect of the project. And then once we have this viral layer of the virus being able to come in and infect the host tissue cells, other members of the team who do a lot with host immune system modeling have added many different cell types and signals that are important for the processes of the immune response. And so, for at this scale, we're using a hybrid combination of the partial differential equations for the signaling factors. So, things like our pro inflammatory cytokines, some specific chemokines, and then and the virions are all modeled with PDEs when they're outside of the cells. And then we use agent-based modeling for the rules and the movement and recruitment of these different cell types. So there are some specific models for interactions in the lymph node. There are others for some specific rules about chemotaxis of various immune cell populations. So again, that was another sub-team, but this is more similar to work that we've done in other contexts. We've built models like this for tuberculosis and other diseases as well. And so this is what it looked like as a simulation before my sub-team joined the project, or we were on the project, but this is sort of the results before incorporating our part. So again, the blue in the background is the healthy epithelium. At the beginning, there was some exposure to the virus at some MOI. And then the other colored cells represent, or colored circles represent different immune cells. The neutrophils are the light blue. You see them very active and sort of spreading across the whole tissue. As the simulation evolves, you start to see more and more red, localized CD8 plus T cells that can kill the infected cells. And so they stay pretty localized once they get there, and they hone toward those areas of infected cells, and they will be responsible for killing. As you watch further, after they've killed those cells, the debris gets cleared out, and this particular model at this stage doesn't have any mechanism for restoring that tissue. So we have some representation of some fibroblasts coming in, but at this point in the model, they hadn't. The animation doesn't show the consequences of the fibroblasts. So that's where my team came in to say, let's have the roles of the fibroblasts in resolving the inflammation and starting to heal that tissue. And these are sort of different simulation versions with different settings for not having the immune system respond to the infection, and so the cells can just die after some amount of exposure, some of the other combinations of immune cell settings. For all of these, we can show the spatial um, domain and at different snapshots in time, the different cell populations. We can also do statistics on that, what's the populations of cells over time, and other kinds of analyses. And so all of those had major tissue loss as a result. And in some cases, people can experience some tissue loss due to COVID infection, but we thought the entire simulation domain being completely depleted wasn't very physiological. So we set out to engage the fibroblasts for being able to have the fibrotic healing. And then in our simulation, we actually don't turn it off. And so it just continues, and we can lead to more of a chronic fibrosis response. And so we think about from the previous parts of the model, the pieces that are very important to us are the action of the CD8 plus T cells killing the infected epithelial cells. And for our part, we say when that happens, there is an anti inflammatory cytokine that gets signaled, and that will then the macrophages will start to shift from being pro-inflammatory to anti-inflammatory at that time. So this action, if these are near each other, will stop the pro-inflammatory signal and will contribute to this anti-inflammatory signal, which then helps fibroblasts to be recruited, embedded fibroblasts to become activated as well, and more deposition of collagen. And these fibroblasts will chemotax towards higher amounts of TGF beta, which is our anti-inflammatory cytokine. So this is one of those simulations of the cell populations superimposed on the chemical field heat map of the concentrations of collagen after one of our simulations. So you'll start to see these types of renderings where the blue part and the other colorful cells come from the first part of the model, and then the fibroblasts from this part of the model, and then you'll see the chemical fields there together. So we, in our Aminal's work, focused on two sources of TGF beta. So when there's damage to this epithelial cell, it can release latent TGF beta. And we by that we mean the TGF beta that is embedded in the matrix, local matrix of the tissue already. And so if the cells die there, it releases the extracellular TGF beta that's already present. So that's our latent TGF beta. And then the other source is anti-inflammatory macrophages that are patrolling the environment in this system, can then also be secreting TGF beta. So we're considering both of those sources, their impact on fibroblasts, and ultimately their impact on collagen. And so we're considering this progression of fibroblasts via this TGF-mediated collagen deposition. So this is the more elaborate flow chart with those rules for how CD8 plus and M1 macrophages interact in a way that shifts towards the M2 phenotype, and the different factors that are secreted. We do have a preprint of this work. It has been submitted for publication, is currently in revisions. So we looked at the different TGF beta sources and the spatial patterns that result. And so we use this notation of um, S without a cross on it. This says that the stationary source is on, and it's the way this is being secreted. This says the macrophages do not secrete the anti-inflammatory signal, and so we get these kind of results where we see these very localized pockets, as imagined, right? If the stationary is the only source, and then it makes sense that when the stationary is turned off and the mobile sources are on, that there's quite dispersed spatial patterns. And here we actually have higher intensity of the collagen that is deposited by having the mobile source. We also looked at different combinations of these. So if the M2 macrophages were high, and then different combinations of the stationary. So high macrophage source, high and low stationary sources. You'll notice that there's not much difference between the red. So here we're looking at the fibroblasts are the main ones that are influenced, but here there's not much different. So the mobile source is dominating this whole thing. There's not really a difference between these two stationary um, cases. And but here now we have the opposite, that the mobile macrophages have a low secretion rate, and then the same two scenarios of the stationary. And in that case, you do see a big difference on what the fibroblasts are doing. You also see some difference between these two now in these stationary cases. So the fibroblasts are the ones that are mostly affected by these kind of different combinations of the sources. And we simulated several different combinations, so stationary only, mobile only, those four different combinations. We also looked at what if the stationary was active for longer, what if the macrophages were active for longer, and the manuscript talks about some different the mathematics of that. And then we looked at amongst our many replicate simulations, I think this is showing 15 replicates, the distribution of those area fractions, right? So we're taking these stochastic simulations that have different behaviors each time, and they all have spatial resolution. We're then doing the statistics on those to calculate things like the area fraction. And so overall, we see, between, no area they all have at least some area fraction of collagen up to about 40%. And these are consistent with the types of experimental observations of 40% chain or 40% fibrotic area fractions in patient populations. So um, it's a little bit different scale than what we're comparing to, but it at least gave us an indication we're on the right order of magnitude here. So we also looked at what if there were some different simulated cases. So one of the simulated cases was that we have our stationary source doesn't get removed as much, and then we have no mobile source. So this is saying that the latent activation is longer. The stationary sources stay there, and so there's a more source. And the consequence of that is that the fibrous outcome is very intense. These are the brightest color that we have of any of the circumstances and that we simulated. And throughout the whole presentation, it's been the same color bar. So these being red, these are the most intense ones we've seen. And they are very localized. So once there's a stationary area, it really just accumulates over time. The other circumstance we looked at is what if we had that, but we also said that the fibroblasts, when they interact with the TGF beta, they don't just respond to it, but they actually uptake that TGF beta. They remove some of the TGF beta from the environment. So our previous simulations just had an interaction. These have a subtraction of the TGF beta from the environment. And the consequence is that we have this fibrotic pattern changes very drastically compared to what it was. The area fractions are modulated. And because there's less of the TGF beta in the environment, you have less recruitment of more of the fibroblasts. So we can explore different mechanistic explanations or mechanistic proposed mechanisms that are maybe difficult to tease out experimentally, but then by looking at these area fractions and types of consequences can help us judge which ones might be more appropriate. So what's next in the story is we've been looking at different patient outcomes that have lung fibrosis differentially. These are some experimental studies that have looked at clinical studies about different patient groups based on sex and age and other sorts of premorbidities and different indications of their the fibrotic area and the resolution of the fibrosis. So we have not yet added to our model the resolution of fibrosis, but we can do that. But we, in this preprint that I point to here, have looked at the impact of different ANG2 or ANG1 receptors. Those are the receptor that the SARS-CoV-2 binds to. And so the connection of that with um, our other models of renin-angiotensin system, et cetera. So really thinking about the um, the receptor density and amount of these things that may influence the fibrosis. And so that's right now just available as a preprint. And then the final story that I'll tell you today is about the Bone Project. And this is work with our fantastic collaborator, Brenda Smith at the Indiana University School of Medicine. She's an expert in bone and dietary interventions and animal models, particularly for mice models. And PhD student Carly Cook is the main student on this project. Aminul Islam as a co-author on some of the work that I'll show today. And then there's other students who've been involved more recently. Um, this work is currently funded by a SUNY seed grant as well as a very recent NIA R21, and we've been supporting it through an R15 and R35 earlier. So in general, for the gut work, or I'm sorry, it's gut, but the bone work, we're thinking about ways that things we eat can impact the immune status of our body. If things we can eat can lead to an immune activated inflammatory phenotype or immunosuppressive, immune tolerant, reducing inflammation type of phenotype. And so, if we sort of the main big picture hypothesis is if these dietary supplements can reduce inflammation in the bone, we think that that would lead to increased bone formation. So in 2018, there's this publication that put together this uh, cartoon model. There was no mathematics here, based on the data that they collected in their study that connected some probiotic that is giving new bacteria to the gut that then would produce more butyrate as a short chain fatty acid. That butyrate was linked to the CD4 plus T cell population and the T regulatory T cell population. So Tregs suppress inflammation and some roles of TGF beta and Wnt10b. So from that part, they linked the Wnt10b to being related to a net bone formation. And they showed a lot of really excellent data in this study. And so we used that as a starting point for building a model that could connect to this kind of first part, kind of whole body dietary supplementation from the gut to the bone. And then in the second part, a detailed model of those impacts on the bone compartment. And so in this model, we were connecting the LGG, which is this probiotic treatment, the butyrate it produces, and the Tregs in the intestine. This is overall a PK PD model. So how does the PK of LGG and butyrate in particular lead to changes in the blood, changes in the bone? And we used symbiology to visualize this, but we actually solved our differential equations in both MATLAB and Python. We made our code also available as a in biomodels type of SBML format as well, just for reproducibility sake. But from a visualization standpoint, we like the way that symbiology represented this. So we have the gut compartment, the blood, and the bone. And we have essentially in each of the compartments the populations of and concentrations of butyrate, the naive CD4 plus T cells that can differentiate to Tregs if the environment is right. And we believe that butyrate is influencing that environment, and as well as changes in the TGF beta and the Wnt production rates. And so we, in the first paper, the IEECR paper, it was really about this whole body and getting it to the Wnt10B that would be in the bone. And in the second paper that appeared in the AICHE journal, both of these are main chemical engineering professional society journals. Um, in the second paper, we detailed the impacts of Wnt on changing the bone remodeling cycle. And I'll tell more about that in a minute. So, bone remodeling is one of these homeostatic processes that our bodies are always undergoing. And we like to think about it from starting at the activation phase, that we're always in this cycle, but initiating the activation phase when there's some damage to osteocytes. These are cells that are embedded in the bone matrix. So we're representing the bone matrix as this gray block. This would be mineralized bone tissue, and we have the osteocytes embedded in there. If there's damage to the osteocyte, which could be a biomechanical injury or stimulus, but also could be chemically modulated, which is how we're interfacing with the gut compartments through a chemical intervention. And they may not be damaged, but stimulated, I'll say. And that triggers the start of a new cycle. And so from there, we consider the pre-osteoclasts and differentiate to osteoclasts that can come in and clean up and consume the cell, resorb the bone, making a region where the pre-osteoblasts differentiate to osteoblasts that come back in. This has a phase of showing reversal cells as well, but here's the osteoblasts that come back in and build new bone through osteoid, sorry, that gets mineralized, but some of the osteoblasts embed into the matrix to form new osteocytes. So in the resting phase, we don't show the process of mineralization because it takes a little bit longer, but that's what's happening in this overall process. So we started from an existing model that does represent the preosteoclasts. It doesn't explicitly show preosteoblasts; they're considered as a kind of steady state population, but there is a source term for the osteo. I guess there is a preosteoblast. Sorry, this one's the one that's constant. Um, so we have the preosteoblastic class, we have the class and the blasts, and the osteocytes, which are represented by an S. This is showing that the class inhibit bone formation, or they actually degrade a bone, and the blasts build back the bone. And the existing model was from a paper in PLOS One by Graham et al. And they had the model tracked the cells involved in bone remodeling. And what we really attracted us to this specific model was that it included the effects of osteocytes. And so osteocytes are not included in some of the more popular classical models. And some of the classical models don't have open source code availability, and so become quite daunting to um, code up if they don't have the cell type you're interested in and don't have code availability. So this one had both simplicity but also had this important role, the osteocytes. And so it does use power law relationships. Some of the other models include not power law, but instead, explicit concentrations of different signaling molecules and things like that. So we're familiar with all the other ones, but this particular one is a power law formulation. So this model was altered in a couple of important ways. So the original model only did one healthy remodeling cycle just to see how the cell types change dynamically. We were very interested in more than one remodeling cycle representing the duration of a treatment, rather than just the impact on a single cycle. We also used previous papers that indicated different hypothesized roles of Wnt's influence on this process. And so we have three ways that we have the Wnt intervening here. Um, that is enhancing um, the differentiation, or the self-replication of the pre-osteoblast, the differentiation from pre-osteoblast to blast, and suppressing the apoptosis of osteoblasts. So these are all now Wnt concentration dependent in a Hill function type of way. And the data in this area is pretty sparse. So we used data from the same lab that had essentially four different studies that they had done. And They had looked at a few different endpoints, but they had looked at when they had knocked out Wnt and when there was a fivefold and 50-fold increase in Wnt expression in these mice. And we use the endpoints as the way to parameterize our model. There's many things that we don't know about the dynamics in between, but this was what we could do with the information available. So we're actually pretty pleased with how the model does predict, even with some of the scarcity of the data that we used for the problem. So these are the predictions of the bone volume changes based on the steady state values of these went fold changes. And then this is a different data set or different lab. And so this is our validation data, pretty good at the 6 cycle version. Our model results continue to rise over time, and it looks like long term biologically that there's some plateau of that. And so we think, in the long term, we should have a mechanism in the model that has this effect get saturated. But so the experiments and things we look at in the future are pretty much in these earlier cycle times, not this long term behavior. Um, so we did connect these things, this whole body PKPD model, and then this more detailed bone compartment model. And again, this was the sort of outline from the published paper that was not about modeling, but about how they saw that their data fit together. And this was our representation of their measured went fold changes. They had the data of these last 2 points, but if we put it in our simulation, here's the dynamic changes in went fold change over time. And if we put that from the first model into our bone model, we get just these tiny bits of change, right? So blue is no change from, or red, yeah, blue is no change. Orange is what happens if we have this amount of butyrate change. Not very much. The data that they presented in Tayeg et al. Was that there would be this like 35 or so percent change, not a 1% change. And so um, our model uses these amounts of went fold change, propagates it through the whole thing, and barely anything happens. So that led us to explore whether there could be a direct role of butyrate. So in the work I've shown you before, butyrate has to influence Tregs, it has to influence um, the TGF beta and the Wnt10B before it can then modulate things in the bone. So we had only added the Wnt effects. Um, there is emerging observations and discussion in various papers that point to maybe there's a direct impact of butyrate. There's not a lot of data toward that. But what we did mathematically was said, well, let's just have a term that could allow butyrate a direct role on every single term, one at a time, every single term in the bone compartment model. And so these are each of the different terms in our process. And then to just test this sort of in this exploration, in silico exploration, we said the coefficient for that particular process needs to make sure that the final endpoint goes through this data point. Right. So these are the curves that result from that process. And again, what's in between is not this is not a proof, but this is more of an exploration. Which of these might be explanatory of this amount of increase? So they're all one at a time. They're not combinations of that. So that's a limitation of this analysis. But what we could do from that is then they all could get to that data point. So that's the first check. Then using that, we then we simulated what were, so that was the data we used for the fitting, the final bone volume piece. But we wanted to simulate also, or in that same simulation, you get the bone volume, but you also get the populations of the other cells, the osteoclasts, the osteoblasts, etc. And so this is just for all of those cases, looking at what's the change in the osteoblast area under curve, osteoclast area under curve, some of the metrics that we use for assessing the reasonableness of the models. And so cases 4 and 5 were outright rejected because these osteoblasts decrease, which is not physiological. So even though it can go through the final data point, the way it does it is non-physical. Um, the 8 and 9 just said, let it change the bone, like let butyrate increase bone volume or let butyrate decrease bone volume without any mechanistic explanation. So we ruled those out. And so it gave us 3 simulated cases that can give us acceptable hypotheses about a direct impact of butyrate. So that means that they're now opportunities for us to go back to the lab with our collaborators and say, can we explore if there's a direct impact on butyrate on osteoblast proliferation, osteoclast differentiation, and osteoclast apoptosis? And so that's sort of part of the ongoing work that we're doing together. So this is kind of one of the first times in our lab that we were able to do hypothesis generation from our in silico studies. So what's next in the story? Carly is continuing as a PhD student looking at the short chain fatty acids influence in normal and estrogen deficient bone remodeling. And so we're specifically um, interested in dietary supplements that are not new biotics, new probiotics, but instead are fiber sources that then can be prebiotic, giving the bacteria you already have in your gut food sources to then ferment and make more short chain fatty acids to suppress inflammation. So we're looking at the both roles of the T regulatory anti-inflammatory cells and the more anti or the anti-inflammatory Tregs and the inflammatory Th17s. And we're doing experiments with OVX and sham operated animals, and we're looking at various diet and immune modulators. And so this is the work with the new R21, as well as some of the work with the R15 and the SUNY seed grant. Additionally, Ariel Leidy is a PhD student who's joined in the last year, and so she's looking at some of the work with the short chain fatty acids and estrogen influence specifically on the gut inflammation. And so, if the gut is already inflamed due to estrogen deficiency, or if it's intact, how might these treatments be modulating local inflammation in the gut? And then does that carry over to systemic inflammation resolution in the bone? So that's most of Ariel's project that she's getting started now. And so I've given you kind of a whirlwind tour of some of the things we work on in the lab, all from a computational and mathematical perspective, lots of differential equations, but a lot of different techniques. You got to see some finite elements, some differential equations, some um, agent-based models, so a variety of different modalities there. And I talked about our bone, lung, and kidney projects that have been going on the last couple of years. But I want to acknowledge again the members of my team. Um, some of the team works completely remotely, and so we only see them when we go to in-person meetings. But this is the crew that was here in Buffalo last fall. So with that, I want to thank you and open the floor for questions.
Thank you, Ashley. That was that was right on time. And yeah, I really liked how you covered different areas and like described this and gives references for all the work that you've described here. And huge fan of you giving a shout out to your students. That's great. Um, all right, so we do have some Q&A. I'll go with the first one. So this question comes from Surjoy. He asks, are there similar models involving adipose tissue modeling in obesity?
You know, I don't know the answer to that. So um, I know that there are some really exciting teams that are working on adipose tissue from an experimental standpoint, and I've been interfacing and seeing some of them on social media or at meetings lately. But and so I could name a few experimentalists that are working on adipose, but I don't know. And for them, my perception is that um, they're really trying to gain traction amongst at least biomedical engineering community about the importance of adipose tissue and its role in obesity and other disorders like lymphedema, et cetera. And that there's a lot of opportunities for those of us that are for more systems biology to collaborate with them and contribute in that space. But I haven't heard of anybody really talking about from a modeling perspective on that side.
Okay, yeah, thank you. Another question comes from Shin Lai, who joined us from Finland. He says, thanks for the great talk. For the COVID model, how did the consortium coordinate modeling tasks among so many groups? And what about the intracellular signaling pathways?
Yeah, so fantastic. So um, one of the biggest things that we did was set up weekly Zoom calls and various technologies like Slack and at GitHub, and we also had this platform called NanoHub where people could, where it would host the GitHub repository and people could like work on it, not work on it, run it like a GUI without having to interface with the code, just kind of check what simulations we're doing, but not have to have like supercomputing resources or a cluster or anything like that. It would just run in the cloud. So we had a number of like infrastructure tools, and early in the stages, we had subgroup meetings. So, my team would meet and talk about the fibrosis and the damage part. The other, like immune or the viral replication team, they would all meet, and we sort of had some pressure for deliverables of like trying to get this done in a timely manner and get it interconnected. We also pre-printed in phases. So, like, okay, here's what's going to happen in the phase version 3 preprint. Let's try to push to get the model outputs to version 3 and then to version 4. We now are on version 5 that's publicly available, aiming to get um, uh, everything cleared up for submitting for publication soon. So there was a lot of interest. I think that helped. Having the Zoom, having kind of clear deliverables on subtasks. I would say now that it's pandemic started in 2020. We're 3.5 years in. There's not a lot of action on those meetings anymore. It's really like some of the students may be meeting and following up with some specific tasks, but the bulk of the work was over. We also kind of set out some guidelines at the beginning about, all right, each subversion gets its own like GitHub piece, and here's how it integrates back into the main part, so that we were version controlling everything, tracking everything from the beginning, that it was open from the beginning. Like the first preprint was done within like a week or so of the project starting, and that we really wanted to be transparent and open and iterative and community spirit, so more people could join, more people could interact. The author list evolved over time, so it really was this spirit of, if you had some relevant computational skills, that you had some experience doing kind of this tissue level agent based modeling, or you knew some of the nuances of the different biological phenomena, your expertise could be integrated within that pretty well. So but Paul did a great job, Paul Macklin, with coordinating that and bringing the team together.
Yeah, and actually, I think the second part of the question was what were there any intracellular signaling pathways that were
Yes.
modeled?
Okay, yeah, great question.
Yeah,
So the viral replication was definitely an intracellular one, where if there's a virion within a certain distance of an epithelial cell, then that triggers this intracellular network that is completely ODE based, based on the viral replication team. Uh, there's another one intracellular that I think is related to maybe some of the explicit chemotaxis rules. I haven't looked into that specific submodel in detail, so I don't know all of the intricacies there. For our work, we don't really have separate intracellular. It's really like, if a certain factor signals a fibroblast, how does that then transduce into something like a fibroblast recruitment rate? And we use some published data about, here's the fibroblast, here's the TGF beta signal it experiences, here's the changes that they observe. So that became more phenomenological rather than a whole intracellular pathway.
makes sense. Uh, all right, yeah, Shin says nice story behind the work, and thanks again for your answers and talk. I just have one question, actually. This was around, I think you gave a shout out to Nick and his finite element modeling work and the glomerular, around the glomerular filtration part of the talk. I was just curious, when you talked about integrating different time scales, how do you take care of that? Um,
Yeah, so it's one that we think about a lot. I don't think we haven't published yet on it. So it's a lot of like, we've got to brainstormed a lot of things that we're in the process of trying.
yeah,
Um, and some of it's going to depend on sort of the separation of the time scales, right? So if you're starting from like, how does glucose oscillate or fluctuate on a daily basis, you're probably not in the same simulation going to track 40 years of patient trajectory for the complete um, virtual twin of their diabetes experience, or at least not with current simulation capabilities, but that could be a future goal. um, so some of it we're thinking about what's maybe if we can connect seconds, minutes, days, hours together in a simulation environment, then we might be able to do that with some coupling of these different modalities. So we have leveraged, or we've looked to some of the cardiac cycle remodeling, cardiac biomechanics coupling that there's a few labs out there who've published on this, and we've talked with some of the authors on what they did. But there's some elegant work that has looked at taking FE which can model the biomechanics, and sort of some of one specific time scale about like how growth can be happening, but then you're letting that run for a certain amount of time and then sending the result to a MATLAB code, having the MATLAB code um, run some update of some sort, calculate a new parameter, have that come back into the FEBio model. And so that could be a way that you're thinking about, the when you make those communications happen is going to really depend on what's the timescales you're interested in and how frequently you need to do that sample. But if some of them really need detailed calculations, but the parameters essentially constant for, let's say, a few hours or a few days, then you like let the change in this influence the other. So there are ways to make those happen more frequently or more coupled, but right now see ourselves using that MATLAB and FEBio approach also because most of our um, chemically based models have been done in MATLAB or Python. FEBIOS just has some nicer connections to MATLAB interfaces. So that's why we're looking towards MATLAB. But it's very much a work in progress. We need to think about for our problem what are the specific timescales of interest, and does it make sense to have them coupled in one simulation, or have a bunch of different scenarios that represent the different states that one could be in, and they're not necessarily dynamically integrated and linked. So we're trying to think about we might do both for different kinds of circumstances.